and I, Sejal, welcome you to our podcast channel Green Gene Talk,
where we talk about everything fascinating in horticulture and plant biotechnology. And today we are diving into a super interesting topic, that is overcoming the pre and post-fertilization barriers in plants and how biotechnology helps us to break those walls.
Exactly, and in simple words, we are going to discuss why sometimes plants don't cross successfully and how scientists use advanced techniques like embryo rescue and pollen tube method to still make it work.
Hi Sejal. So let's start from the basic in plant breeding. That is, we often want to crosses between two different species or varieties to get new traits for the better yield or fruit quality purpose. So what is your opinion in this matter, Sejal?
Right, Risita, but sometimes the plants say no, not compatible. These are what we call reproductive barriers, which are mainly of two types: pre-fertilization and post-fertilization barrier. Rishita, can you further explain about them?
Yes, Sejal, of course. Pre-fertilization barriers are the barriers which prevents the fertilization from happening, and these can be spatial, temporal or mechanical in nature. This occurs when the plant fails to germinate the pollen, or even when the plants flower at two different times.
Exactly. For example, in citrus and grape, pollen stigma incompatibility is quite common. Sometimes the pollen tube fails to grow properly through this type.
Yes, Sejal, you are correct. Post-fertilization barriers are occurs after the fusion of gamete. The embryo abortion or even the endosperm failure or the hybrid sterility can cause this.
Yes, like in the case of Vitis vinifera and wild species, the embryo often aborts early because the endosperm doesn't develop properly.
So why are these barriers a big deal? Because they prevent from interspecific and intervarietal hybridization?
Exactly. Imagine if you cross disease resistant from a wild species with a sweet fruit quality of a cultivated one. If natural fertilization fails, your breeding program stops right there.
You are absolutely right, Sejal. This is where biotechnology comes to the rescue.
Modern biotechnology offers several techniques to overcome these barriers. Some of the key ones includes pollen tube pathway method, embryo rescue, in vitro fertilization and somatic hybridization. So Rishita, let's talk about pollen tube pathway method.
Yes, why not? The pollen is placed directly on the stigma, and after the pollen start growing, a small amount of fertilization medium, nutrient and growth medium is given to them and introduced into the ovary through the style.
Right. This helps the pollen tube reach the ovule, bypassing incompatibility reaction that might otherwise stop fertilization. It's especially used in the crops like lilium and brassica. Now, the most famous one, embryo rescue. This technique literally saves the young embryo before it aborts. The immature embryo is excised and grown on a nutrient media under sterile in vitro conditions.
Yes, it allows hybrid embryos which would normally die due to the post-fertilization barriers. It's widely used in grape, citrus, apple and many more interspecific hybrids in India, like tomato. For example, in citrus breeding, the cross between sweet orange and trifoliate orange results in embryo abortion. But with embryo rescue, viable hybrid plants can be obtained. And sometimes when even embryo rescue fails, breeders use ovulate horticulture instead of that. That is even the earliest stage of rescue.
So now, let's talk about somatic hybridization or protoplast fusion. Here, instead of relying on sexual reproduction, scientists fuse protoplast, which is plant without cell, from two different species.
Yes, the fusion of protoplast can regenerate the hybrid plants, getting genetic material from both the parents. This technique is great for overcoming the sexual incompatibility barriers. For instance, in citrus, somatic hybridization has been used to combine cold tolerant and disease resistant from wild species with high fruit quality of cultivated ones. So let's talk about another approach, that is in vitro pollination and fertilization, where the pollen grains or the pollen tubes are placed directly onto the ovule or ovary under lab condition. So Sejal, what is your opinion in this?
Yes, this methods help in studying fertilization mechanism and also assist in cases where natural fertilization is blocked. For example, due to stigma style incompatibility, and it's often a preliminary step before embryo rescue, especially in wide hybridization experiments. To make this more relatable, let's talk about few success stories. In tomato, embryo rescue has been used to transfer disease resistance from wild species like Solanum peruvineum into cultivated varieties.
Yes, in grapes, embryo rescue and ovule culture have been key to developing the hybrid between seedless and seeded varieties. Even in citrus, protoplasm fusion had led to somatic hybrids like sweet orange plus trifoliate orange, giving plants with better rootstock traits.
Yes, even in Brassica, the pollen tube pathway method helps to produce interspecific hybrids between Brassica campestris and Brassica oleracea, which could not be done naturally. So Rishita, let's just wrap the podcast. We can say pre and post-fertilization barriers can be tough, but biotechnologies has given us the tools to overcome them and unlock genetic diversity.
Yes, techniques like pollen tube pathway, embryo rescue and protoplasm fusions are like the breeze between species, helping us to create better and more resilient crop for the future.
That's all for today's episode of Green Gen Talks.
We hope you enjoyed this talk. If you did, don't forget to
Like, follow and share.